Welcome to topic 3.2 of module 3. In this particular topic we will learn about growing and identifying bacteria. During the course of this video, try and answer as many questions as you can from module 3, topic 3.2. The learning objectives for this topic are to describe where microbes acquire energy and carbon sources, describe the growth conditions of different microbes, including their physical and chemical requirements, and to describe ways to measure microbial growth and the reasons for using each method. For an organism to grow, it must acquire energy and carbon from different sources. Organisms that derive energy from light are known as phototrophs, while organisms that obtain energy from chemical reactions, such as oxidation, are known as chemotrophs. Autotrophs are organisms that can fix carbon from the atmosphere, particularly CO2, while heterotrophs require carbon from organic compounds. Photoautotrophs are organisms that acquire energy from light and carbon from atmospheric CO2. Common examples of photoautotrophs are plants, algae, and photosynthetic cyanobacteria. Photoheterotrophs acquire energy from light. However, they are dependent on organic compounds for their carbon source. Green and purple bacteria are the most common photoheterotrophs. Chemoautotrophs are organisms that obtain energy from oxidation of inorganic compounds such as sulfur, magnesium, and iron, but obtain their carbon from atmospheric CO2, as seen in some bacteria and archaea. Organisms that completely depend on organic compounds for energy and carbon are known as chemoheterotrophs. Animals, protozoa, and some bacteria fall into this category. For microorganisms to grow, a set of conditions, both chemical and physical, must be met. The chemical elements that are necessary for growth are carbon, nitrogen, sulfur, and phosphorus. These compounds play an important role in the synthesis of DNA, amino acids, carbohydrates, and lipids. Physical growth requirements include temperature, pH, and the osmotic environment. Oxygen is required for incorporation into organic molecules, and oxygen makes up a large proportion of compounds, for example DNA and amino acids. It is used in respiration aerobes. However, not all microbes require oxygen to breathe. Some organisms can breathe without oxygen, and these ones are known as anaerobes. So, can anaerobes infect humans? Well, the human body is fairly oxygen rich because we have a good network of capillaries that deliver oxygenated blood throughout the body. However, anaerobic microbes can still be seen in deep wounds and internal organs where there is little oxygen supply, usually in areas of reduced blood supply due to damaged blood vessels or blood vessel disease. Some examples of anaerobic infections include gas gangrene, tetanus, and a few dental infections. Respiration using oxygen can generate highly reactive byproducts known as reactive oxygen species. Aerobic bacteria have enzymes that can neutralize these reactive oxygen species. However, because anaerobic bacteria lack these enzymes, respiration using oxygen can harm these cells. The table in this slide is an illustration of the effect of oxygen on the growth of various types of bacteria. In the middle here we have 5 test tubes with a solid agar medium, with all the necessary chemicals that a bacteria needs to grow. Oxygen is present in the gaseous compartment of the tube at the top. The oxygen can only diffuse approximately 1 to 2 millimeters into the solid medium below. Usually, an aerobic bacteria would grow on top of the solid agar medium because it requires oxygen. Anaerobic bacteria would grow at the bottom of the tube because the environment at the bottom of the tube does not have oxygen within it. Obligate aerobes only grow in the presence of oxygen, which is on the surface of the agar medium. Aerobic bacteria have enzymes such as superoxide dismutase and catalase, which can neutralize toxic forms of oxygen that are produced as byproducts of respiration. Facultative anaerobes can grow in the presence and absence of oxygen, However, greater growth can be seen in the presence of oxygen. E. coli is one of the best examples for a facultative anaerobe. Obligate anaerobes can only grow in the absence of oxygen because these bacteria lack the enzymes that can neutralize harmful forms of oxygen. Aerotolerant anaerobes require anaerobic conditions to grow. However, as their name suggests, they can survive in aerobic conditions as well. This is due to the partial presence of protective enzymes. The microaerophiles only grow specifically when oxygen concentration is low. Under normal conditions, these organisms can produce high levels of toxic substances that can harm the organism. Carbon, nitrogen, sulfur, and phosphorus are the chemical growth requirements for bacteria to grow. These compounds act as the basic building blocks for biological molecules. Autotrophic bacteria acquire carbon from inorganic sources, such as atmospheric CO2, while heterotrophs acquire carbon from organic compounds, such as other organisms, plants for example. Most of the nitrogen comes from organic matter, such as proteins. However, inorganic sources of nitrogen can be ammonium ions, which is usually found in soil. Some bacteria, called nitrogen fixers, are in a symbiotic relationship with plants such as legumes. These bacteria fix atmospheric nitrogen and make ammonia from it. Sulfur and phosphorus are obtained from organic sources or soluble ions, such as phosphates and sulfates. Temperature is one of the most important physical growth requirements for bacteria to grow. The graph in this particular slide shows the rate of growth of various bacteria at different temperatures. The peaks in the graphs represent optimal growth temperatures for different bacteria. For example, mesophiles grow between 10 degrees and 40 degrees Celsius. However, the bacterial growth is highest at roughly 37 degrees Celsius, which is our normal body temperature. Thermophiles and hyperthermophiles thrive at higher temperatures, while psychotrophs require lower temperatures to grow. Optimal temperature is the temperature at which the rate of growth is the highest. Bacteria have specific pH requirements as well. Some may survive in low pH conditions, such as the acidic conditions inside our stomach. Helicobacter pylori is a stomach bacteria that is responsible for causing peptic ulcers. However, most bacteria grow around a pH of 7, or neutral pH conditions. Maintaining optimal osmotic environments, which is the balance between water and ions, is crucial for the survival of bacteria, because water can move to an area of high solute concentration within the bacteria through osmosis. However, some bacteria have specially developed cell walls and mechanisms that allow them to grow in hypertonic, photonic conditions. The following figure shows bacteria when placed in an isotonic solution. The cell survives and grows because the solute concentration inside the cell is equal to the solute concentration outside the cell. Hence, the cell does not lose or gain water. However, if the cell is placed in a hypertonic solution, because the solute concentration outside the cell is much higher, water from inside the cell will move out, inhibiting cell growth. Escherichia coli is one of the most studied bacterium in the world of microbiology. As described earlier, E. coli requires specific growth conditions to grow in the lab. They require a nutrient medium that is extracted from yeast and has a perfect balance of nutrients such as carbon, sulfur, and nitrogen. It also grows fastest at 37 degrees Celsius, which is not surprising as E. coli can be found inside the human body. Other physical requirements include that the medium be at a pH of 7 and a saline concentration of 0.9%. All of the above mentioned physical and chemical conditions must be met to culture this bacteria. However, we still cannot culture more than 99% of all bacteria because we don't know their optimum growth requirements. Agar, the gelatinous substance extracted from seaweed, is used as a medium to grow bacteria. The bacterial growth medium can be either solid or liquid in nature, depending on the amount of agarose present. This growth medium can be used to isolate and identify bacteria. Selective media are specially prepared growth mediums that support and allow the growth of only one kind of microbe and inhibits undesired organisms. Selective media usually contains antimicrobials, alcohols, or dyes that can prevent the growth of non-desirable species of microorganisms, whilst isolating target organisms from a mixed culture. Differential media are mainly used to distinguish bacteria based on growth characteristics and biochemical properties, such as a change in the colour of the growth medium surrounding the organism. Differential media is prepared by adding compounds such as horse blood to the medium. Bacterial growth generally refers to increasing numbers of bacterial cells. But why are we interested in bacterial population growth? It's to understand the spread of infections and to know the bacterial load, which is a measurable quantity of bacteria in an organism. Knowing bacterial load can play a key role in the treatment of an infection. And also it's important for the production of compounds. Bacteria divide by binary fission. In terms, binary fission means a single cell splits into two cells. In the first step of binary fission, the bacterial cell elongates and the chromosome inside is replicated. Once the genetic material is separated and sent to opposite ends of the elongating cell, a division ring forms in the middle of the cell, cleaving the cytoplasm and the cell wall into two, eventually leading to the formation of two new daughter cells. The fundamental thing to remember here, every cell gives rise to two new cells. Therefore, the growth of bacterial cell populations is exponential. However, this is dependent on the availability of nutrients. Under favourable conditions, bacteria can double its population depending on the time it takes to divide. This growth in population is by geometric progression, which means if you have two cells, you get 4 daughter cells. From 4 you get 8, and so on. This is called exponential growth. The population of bacteria after a number of divisions can go from 2 cells to 1,000 cells. For example, E. coli takes 20 minutes to divide, which means once every 20 minutes, E. coli can double its population. The exponential growth in bacterial populations can be calculated using the formula 2 to the power of n, where n represents the number of divisions that have occurred. For example, if a bacteria has undergone more than 40 divisions, its population would be more than 1 trillion cells. The time it takes for a bacterial population to double is called the generation time. It is very important to know the generation time of a bacteria, because it can vary between different species. For example, Clostridium perfringens, a bacterial agent responsible for gas gangrene, has a doubling time of 10 minutes, making it one of the fastest multiplying bacteria. However, bacterial growth is not infinitely exponential. Bacteria will stop dividing if the necessary physical or chemical growth requirements are exhausted. There are different growth phases in a bacterial population. When a flask with fresh medium is inoculated with a given number of bacterial cells, the cells initially will take a little time to adjust to those new conditions. This is called the lag phase. The cells will then start to divide exponentially in the log phase or exponential growth phase. In this phase, the number of new cells is greater than the number of cells dying. The exponential growth phase in the population of bacteria continues until they reach the stationary phase. In the stationary phase, the nutrients in the medium start depleting and bacterial growth slows down. In this phase, the number of new cells is equal to the number of cells dying. Eventually, the cells will enter the death phase or the logarithmic decline phase, where the growth medium does not have any more nutrients to support bacterial population, and the bacterial cells start to die. In the death phase, the number of cells dying is greater than the number of new cells being formed. Bacterial population can be counted using either the direct or the indirect method. In the direct method, a sample, or the dilutions of a sample, are inoculated onto the agar plate. After the bacteria have grown exponentially, the number of colonies on the plate is counted and multiplied by the dilution factor to elucidate the bacterial count in a milliliter of a given sample. In cases where the sample with bacteria is too dilute, it can be concentrated using filtration first. Other techniques include direct microscopy, real-time PCR, and flow cytometry. However, these techniques can be very expensive. Alternatively, indirect techniques include using turbidity, absorbance of light, metabolic activity, and measuring mass. Counting bacterial populations using indirect methods will be further explained in your tutorial sessions. From this video, we have learnt about the different sources from which organisms acquire energy and carbon. We've learnt about oxygen-dependent and independent bacteria, the different chemical and physical growth requirements of bacteria, how bacteria divide and the different growth phases of bacteria, and finally, we have also covered how to count bacteria using both direct and indirect methods.